Hello everyone, good evening and welcome back to the probably last session of biochemistry for this series. We'll try to wind up the story today. So let me just confirm from you that you're able to see the video, you're able to hear the audio, and you can see my screen clearly. Please confirm to me that all of these are okay. Probably you would not be able to see my screen. I'm switching it on, and now you should be getting the screen. I hope. There's the screen. Yeah, there it comes. The screen, and and yeah, you should start getting the screen too. Don't worry. So here goes the obvious. Here's thing. Maybe later. Later. By plus. Yes. Okay. So friends, just let me know that you are able to hear me, you are able to see me, and you are able to see my iPad screen. Just please confirm these. I presume everything is working fine. Okay. Righto. So yesterday we were doing replication and we continue with the same story. Let's see. Very key much. Okay. Alright, great. So just to ensure that everything is working fine, are you able to see the video? Are you able to see the screen, or are you able to hear me? Great. So we'll continue from where we left yesterday. Very good. There is a question, sir. The leading strand had no primer. The leading strand had no primer. I'll show that diagram again. The leading strand had no primer. That was a very important observation. The leading strand had no primer. See, this is the diagram that we had. The leading strand did not have any primer. How do you catch these things? Very good. Okay. So please note, by the time we reach here, by the time you reach here, the Okazaki fragment on the second half of the replication fork, or you can say the second replication fork, will have processed and progressed to merge with the strand which we call the leading strand, and thereby giving us a picture like this. I've increased this thickness so that we can appreciate it better. To give us a picture like this. You know that the Okazaki fragment, the primer part, is replaced by combining with the next Okazaki fragment. So for this scenario, for the leading strand, the Okazaki fragment on the second replication fork is the next Okazaki fragment. So it will proceed and it will have replaced the primer of the leading strand. By the time we reach here, by the time we are able to see this picture, the primer of the leading strand has been replaced. That is why when we draw the diagram of the replication fork and we are trying to show a little bit of extra DNA, in that case the primer of the leading strand will already have been replaced. That's why generally in the diagram you will not see the primer on the leading strand. The leading strand is already merged with the Okazaki fragment on the second replication fork, and the primer part has already been replaced by the DNA. That is why the primer will not be seen. I hope that is clear. So let's continue. I told you that we have finished talking about the proteins involved in the replication, right? And now we'll talk a little bit about the enzymes of replication, how the prokaryotic enzymes compare with the eukaryotic enzymes, and then we'll talk about DNA damage and the DNA repair. Let's take it back there. Yeah. Okay. So now quickly we'll compare the prokaryotic the prokaryotic polymerases with the eukaryotic polymerases and compare their respective functions. Actually, I hope you got it. Great, great. That's so nice. So let us quickly compare this. Alright, here we go. The prokaryotic, and this will be the eukaryotic. Right. So remember, in the prokaryotes we had the polymerase 1, polymerase 2, and polymerase 3. What I'll do is I'll replace the headings. Rather, I'll make this the function column, and I'll make this the eukaryotic column. So it will be simpler for us to compare. Okay, this will be simpler for us to compare. So polymerase 1 we had seen is responsible for replacing the primer. It is responsible for replacing the primer. At the same time, it is also playing a role in a repair. It has a role in a repair. The polymerase 2 only has a role in DNA repair, whereas polymerase 3 is primarily responsible for the polymerization. It is primarily responsible for polymerization of leading and lagging strands. The primary polymerization will be done by the polymerase 3, and after that the primers can be replaced by the polymerase 1. In addition, we said that there is the primase here, and the primase is also known as DNAG, primase known as DNAG protein, and it will synthesize the primer. Please note, the primase in the prokaryotes is an independent protein. It is an independent protein. So polymerase 1 has two functions. It replaces the primer and it has a role of DNA repair. Polymerase 2 only does the DNA repair. Polymerase 3 primarily causes the polymerization of leading and the lagging strand. In addition, we have the protein called primase, which is a protein called DNAG, and it is an independent protein which synthesizes the primer. The primer, if you recall, is RNA in nature. It is RNA in nature. So these are the functions of the prokaryotic proteins involved in the polymerases involved in the replication. Let's compare these functions with the eukaryotic cell. So here The primer is replaced by, I can't help it. The primer is replaced by the DNA polymerase beta. Replaced by polymerase beta. In eukaryotes, I told you we have the alpha, beta, delta, gamma, epsilon, like that. But it doesn't have the repair function. It doesn't have the repair function. The polymerization of leading and the lagging strands done by delta and epsilon, done by the delta and epsilon. So they will synthesize the leading and the lagging strand. They will synthesize the leading and the lagging strand. The primer is synthesized by primase, which in turn is part of, it is not an independent protein, it is a subunit, part of polymerase alpha. So alpha, beta, delta, epsilon. What happens to gamma? So let's see what happens to gamma. We have the polymerase gamma. All of these are polymerases, please note. All of these are polymerases. So polymerase gamma, this is the mitochondrial DNA polymerase. Please note, obviously the mitochondrial DNA polymerase is not required in the prokaryotes. It is not required in the prokaryotes because it doesn't have the mitochondria, whereas it is required in the eukaryotes. So we have the additional polymerase gamma in the eukaryotic cell. So I hope this will clarify the picture that we are talking about. It will clarify the picture that we are talking about. The functions of the prokaryotic and the eukaryotic polymerases. The functions of the prokaryotic and the eukaryotic polymerases. Pol 1 similar to polymerase beta. Pol 3 similar to delta and epsilon. The primase is a subunit of polymerase alpha in the eukaryotes, and the polymerase gamma that we have functions as the mitochondrial DNA polymerase. Now here I'd like to specify one thing. In the eukaryotes, the number of polymerases is much more than what I have mentioned. They are much more than these 5 I have mentioned, but these 5 are the primary counterparts of the polymerases as we see in the prokaryotes. It doesn't mean that they are not other polymerases. It doesn't mean that other polymerases do not participate. They are there, they participate, but their function is not major, and therefore they are not discussed at your level. In addition, there will be a large number of other proteins in addition to the polymerases which participate in the process of replication. However, they are not mentioned. This story will repeat always in the transcription as well as in the translation also, where we will not mention the minor proteins, but remember the large number of proteins and enzymes are involved in these activities. However, we are only highlighting the major ones. In addition, these procedures are still under investigation, and it is possible in the next edition some changes in the information may appear. Alright, so we have to be mentally prepared that okay, new editions might bring some changes. These information are not absolute, they're continuously changing information, and therefore we have to be alert about updates. But there are updates which I get to know, then obviously I'll be posting them and sending them across to you. Okay, so this is about the polymerases, and now we move on to the DNA damage and the DNA repair. If you have any questions so far, please send them across. Now we are going to the DNA damage first, and then we talk about the DNA repair. When we talk about a DNA damage, you have to understand the DNA damage is a continuous process which is occurring in all the cells, and to counter this DNA damage, the cells have the DNA repair mechanisms. So first we mention the common types of DNA damage and what are the corresponding repair mechanisms which are employed to counteract those DNA damages. Sometimes the DNA repair mechanisms are not working properly, in those instances we have specific associated conditions when a particular DNA repair mechanism is not working. So we'll list them also, and they're quite frequently asked in your exam. So let's see the types of DNA damage. First, involving a single base. The DNA damage involving single bases. Here you can have mismatch. When you're doing the replication, you are supposed to pair A with T, G with C, C with G, T with A, but sometimes in the hurry there can be some mistake. Instead of T in front of A, you put up C, so that's a mismatch. So that is called the mismatch. And sometimes there can be spontaneous deamination. Spontaneous deamination. Very commonly, the result of this spontaneous deamination is the conversion of the cytosine to USL. You remember when we are doing the pyrimidine synthesis, the UTP is converted into CTP. So the backward reaction can occur when there is a lot of free energy in the system, for example from the UV rays, from chemical reaction. If there is free energy, the backward reaction can occur, which is known as spontaneous deamination. Rarely there can be conversion of adenine to hypoxanthine. Rarely there can be conversion of adenine to hypoxanthine, but more commonly we see the conversion of cytosine to uracil. So these are single base damages. Then under the heading of the two base damage, commonly what you get to see are the thymidine dimers. You get to see the thymidine dimers, right? And lastly, you can have what are known as breaks in the DNA. Breaks in the DNA. These can be single strand DNA breaks, or they may be double strand DNA breaks. So single strand DNA breaks and double strand DNA breaks. So these are some of the common types of DNA damages that will come across. Single base, where mismatch or spontaneous deamination can occur. The two base, which commonly presents as thymidine dimers, and involving the DNA breaks. Sometimes a single strand will break, sometimes both the strands can break. Looking at the repair mechanisms, for mismatch, we simply have what is known as mismatch repair. We simply have what is known as mismatch repair. For spontaneous deamination, we have the repair mechanism known as base excision repair. The repair mechanism commonly known as the base excision repair. For thymidine dimers, we have the nucleotide excision repair. The nucleotide excision repair. For the single strand DNA break, we have the homologous repair. The homologous repair can also occur for double stranded DNA breaks. Please note, the homologous repair can also occur for double strand DNA breaks. However, it is rare as a mechanism for double strand DNA breaks. It can occur, but it is not commonly employed. What is commonly used is known as non-homologous end joining, meaning the actual repair doesn't occur, only the two strands which have broken off are brought close to each other and they are joined. This is known as non-homologous end joining. The non-homologous joining is written as NHEJ. The homologous repair is HR. The nuclear excision repair is NER. The base excision repair is BER. And the mismatch repair, commonly written as MMR. So we have the mismatch repair, the base exchange repair, the nucleotide exchange repair, the homologous repair, and the non-homologous end joining for the corresponding damages that we have seen. Now I told you sometimes these repair mechanisms may be defective when they are associated with corresponding specific disorders. So we are going to match them with their corresponding specific disorders. When the mismatch repair is not working, in that case we get what is known as the Lynch syndrome. We get what is commonly known as the Lynch syndrome. The Lynch syndrome is also known as hereditary non-polyposis colonic carcinoma. It is also known as the hereditary non-polyposis colonic carcinoma. Now, the base excision repair, when it is defective, causes a condition called mute Y homologue associated polyposis. The short term which is used is mute Y homologue associated polyposis. It is a subtype of the better known familial adenomatous polyposis. It is a subtype of familial adenomatous polyposis. Okay, so the mismatch repair associated with the Lynch syndrome, also known as the hereditary non-polyposis colonic carcinoma. The base exchange repair associated with the familial adenomatous polyposis. The genetic diagnosis is mute Y homologue associated polyposis. The nucleotide exchange repair can be associated with multiple disorders, but the most commonly seen is the Xeroderma Pigmentosum. The Xeroderma Pigmentosum. We also have what is known as trichothiodystrophy. And lastly, what is known as the cocaination. The spectrum of disorder, all of them have similar presentation, that is photosensitivity and frequent skin lesions. The Xeroderma pigmentosum is different from the others because in Xeroderma pigmentosum there is higher sensitivity for conversion of skin lesion to the cancer. Alright, in the other two the tendency for cancer formation is comparatively less, but in Xeroderma pigmentosum the tendency for cancer formation is significantly higher. That is why it is important and is very frequently asked in your exam, so you should be careful about that. The Xeroderma pigmentosum which presents with the photosensitivity and skin lesion. On doing the histopathological examination, very frequently the skin lesions turn out to be malignant in nature. The homologous repair associated with Bloom syndrome, the Werner syndrome, and defective BRCA1 and 2 genes. Defective BRCA1 and 2 genes. These two genes are important because you know very well that they are associated with breast cancer. They are associated with breast cancer. Now, if only BRCA1 or BRCA2, if only one of those genes is defective, then we get the breast cancer. However, if both of them are defective, then there is a very high chance of breast cancer with ovarian cancer in females and breast cancer with prostate cancer in the males. So remember, only one gene defective, breast cancer. Both of them defective, then ovarian / prostate will also be there in the males. Defect in non-homologous end joining presents with two SCIDs. One is simply the SCID and a variant known as radiation sensitive SCID. So there are two variants of SCID which we may see in the non-homologous end joining. So obviously they cannot be accommodated on the same screen, so I'm decreasing the size so that you are able to see it. Okay. Okay. First we have the mismatch repair. The repair mechanism, mismatch, and the repair mechanism is the mismatch repair. Then we have the conversion of cytosine to uracil. Repair mechanism is the base excision repair. Then we have the thymidine dimers. The repair mechanism is the nucleotide excision repair. Then we have the single strand DNA break. Repair mechanism is the homologous repair. We have the double strand DNA break. Repair mechanism is the non-homologous end joining. Coming to the associated disorders, the mismatch repair is associated with the Lynch syndrome, also known as the hereditary non-polyposis colonic carcinoma. The base section repair is associated with the type of familial adenomatous polyposis. The very specific name is the mut-Y homolog associated polyposis, also known as MAP. Mut-Y homolog associated polyposis, also known as MAP. The nuclear acid repair is associated with the spectrum disorders, the Xeroderma Pigmentosum, most important, the trichothiode dystrophy, and the Cockayne syndrome, where we have the skin lesion, photosensitivity, and very frequent conversion to malignancy in case of Xeroderma Pigmentosum. The homologous repair is associated with the Bloom syndrome, the Werner syndrome, and defect in the BRCA1 and the BRCA2 gene, which is very strongly associated with development of breast cancer. If both genes are defective, in females we will also have the ovarian cancer, and in males we will have the prostate cancer. A defect in NHEJ will give rise to the severe combined immunodeficiency or the radiation sensitive severe combined immunodeficiency. So these are the very important DNA damage and the repair mechanism table. Please note, at least one question is going to land up from this table. This is a very important table. So if you have any doubts, if you have any confusion, anything you want to ask from this table, please feel free to do so. You must know this table. You will have to remember this table thoroughly, because very frequently it will land up in your exam. Alright, so that is about the DNA damage and the DNA repair mechanisms. Now I'll quickly move on to the transcription. This we have completed the DNA replication, the DNA damage, and the DNA repair. Now we are moving on to transcription, which is the RNA synthesis. Transcription, which is the RNA synthesis. Right? Any questions so far from the DNA segment that we have discussed, from the synthesis of DNA and the repair mechanisms? The agents which are going to cause the prevention of the replication, because that segment will be separately covered in the chemotherapy in your class. So I'm not going into the details of the agents which block the replication. Alright, they will be separately covered in the topic chemotherapy in the pharma class. Okay, moving on to transcription, also known as RNA synthesis. Now transcription is very simple. It is achieved in stages. It is achieved in stages. We describe the stages as initiation, elongation, and termination. We'll see something similar when we are doing that translation, that is protein synthesis. So we'll notice something similar stages will be there. Let's look at the three stages that we've described for transcription, that is RNA synthesis. Here I'll repeat again, Remember, in prokaryotes, we have one polymerase, one RNA polymerase in prokaryotes. Whereas in eukaryotes, we had mentioned the three RNA polymerases. The three RNA polymerases in the eukaryotes. So in prokaryotes, we have a single RNA polymerase, and eukaryotes, we have at least three RNA polymerases, labeled as polymerase 1, 2, and 3, which we have previously seen. So let's talk about initiation. We are doing this discussion again in the prokaryotes. We talked about the replication in the prokaryotes, and now we're talking about the transcription also in the prokaryotes. Now, the RNA polymerase in the prokaryotes is having multiple subunits. It has two copies of alpha, one copy of beta, one copy of beta dash, and one copy of the omega subunit. These are the 5 subunits in the RNA polymerase of the prokaryotes. Now this cluster is transcriptionally active, means it has all the functionalities by which the transcription can occur, but it is missing one very important thing, and that is from where we have to start the transcription. It is unable to identify the site from where we can start the transcription, and for that it needs the sigma subunit. Without the sigma subunit, it is unable to identify the position or the location from where the transcription must start. So until the sigma subunit is there, we don't get what we had previously said as the holoenzyme. So when the sigma subunit is there, we get the RNA polymerase holoenzyme, means the finally fully functional enzyme. The RNA polymerase holoenzyme is formed. This RNA polymerase holoenzyme will now go and attach to the DNA. It will go and attach to the DNA, and it will start scanning the DNA. It will start scanning the DNA for what? For a specific sequence, which is called the promoter. It will start scanning the DNA for the specific sequence, which is the promoter. In this context, remember, in prokaryotes, we have specific sigma subunit for the specific promoter. So to which promoter the holoenzyme will go and attach depends on which sigma subunit is attached to the RNA polymerase holoenzyme. So this whole complex will go and attach. This whole complex will go and attach with the DNA, where the sigma subunit finds the counterpart promoter sequence. This means in the RNA polymerase holoenzyme, only segment which is able to attach to the DNA, the only segment which is able to attach to the DNA is the sigma subunit. The sigma subunit attaching to the promoter. So first point to note, the RNA polymerase holoenzyme will go and attach to the promoter site which corresponds to the sigma subunit. Once this attachment occurs, there is a denaturation of the overlapping segment of DNA. Whatever DNA is covered by the RNA polymerase holoenzyme, that segment is separated, and only that segment is separated. Following the separation, there is synthesis of a small segment of RNA. By reaction of RNA is synthesized. Now, please note, previously the polymerase cannot attach to DNA anywhere except the promoter site, because the sigma subunit is able to identify the promoter site, but other segments are unable to identify any site on the DNA. But once you synthesize the RNA, each of these positions can be identified by the RNA polymerase, because RNA polymerase has itself put in those RNA nucleotides, and therefore They are providing the attachment to the RNA polymerase, with the help of which the RNA polymerase can now start moving forward. The RNA polymerase can start moving forward along the DNA. Please note, during this movement forwards, the RNA polymerase will not be attached to the DNA. It will be attached to the RNA. During the forward movement along the DNA, it will move along the DNA, but it will be attached to the RNA because it doesn't identify any other segment on the DNA. Only segment which was able to identify was the promoter, which has been left behind, and therefore this step which the RNA polymerase takes by moving forward after the synthesis of the small segment of RNA is known as promoter clearance. As you can appreciate, now it is able to leave the promoter behind and start moving forwards along the DNA. That is why this is known as the promoter clearance. Promoter clearance is the last step of initiation. Once we have the promoter clearance, then we have entered the second phase, which is the elongation. Elongation is very simple. The RNA polymerase will keep moving forwards, and it will keep synthesizing the RNA by attaching the complementary bases to the sequence in the DNA. In this manner, our RNA will keep increasing in length. So nothing special about the elongation phase. What we will talk about is the termination. About termination, you have to remember the termination can occur when one of the two conditions is fulfilled. Termination will occur when one of the two conditions is fulfilled. First condition is the transcription of Rho binding site. When the Rho binding site is transcribed, then the termination can occur. This particular manner of termination is known as Rho or ATP dependent termination. Rho because Rho factor will be used, and ATP because ATP is consumed in this process of termination. So when the Rho binding site is transcribed, we have what is known as the Rho or ATP dependent termination. The second mechanism by which the termination may occur is by fulfilling the two conditions. Transcription of self complementary segment. So in this termination, first we have the transcription of the self complementary segment, followed by, this segment must be followed by transcription of poly U segment. Remember, U is the counterpart of T, and therefore it will attach with A, and the pairing between A and U will be 2 hydrogen bond per pair, and therefore this is an area of weakness, or it is the weak segment. In this case, when these two conditions are fulfilled, we don't use the Rho factor. ATP is not consumed, and therefore this is known as Rho or ATP independent termination. Please note the details of the two processes are not required. What we need to know is we have the two different processes of termination. One is the Rho or ATP dependent termination. The criteria for this termination is during transcription we should be able to transcribe the Rho binding site. This is where the Rho will come and get attached. Using the energy provided by ATP, it can terminate the transcription. The second mechanism is the Rho or ATP independent termination. For this mechanism to come into play, we have to fulfill two conditions. Those two conditions are, first, the transcription of a self complementary segment, and this should immediately be followed by the transcription of a poly U segment. Once these two are completed, then quickly we will have the Rho or ATP independent termination. Please note, in the self complementary segment, this is So this is how the termination is occurring in the prokaryotes. In eukaryotes also it is quite similar. One very important point to note for the eukaryotes is the sigma subunits. Rather, what we have are the transcription factors. What we have are known as the transcription factors, written as TF, and they are numbered as 1, 2, 3, 4, 5, 6. They are numbered like that, the transcription factors. The most important transcription factor happens to be what is known as transcription factor 2D. It is the most important regulatory molecule in the transcription of the eukaryotic cell. In the eukaryotic cell, the most important regulatory molecule happens to be the transcription factor 2D. The transcription factor 2D is itself said to be made up of two important components. One is, in the promoter site in the eukaryotes, we have a specific sequence called TATAAG, which is known as Tata box. You don't need to know the sequence, just remember there is a sequence labeled the Tata box. It can be anywhere upstream in the promoter sequence, between 9 to 35 nucleotides upstream it is there. So we have a protein which will identify this Tata box, and therefore it is known as the Tata box binding protein, or simply the TBP, the Tata box binding protein. Oh my god, I'm so sorry. Let's see, why is it not visible? Just give me 1 minute. Let me sort out this connection issue. There is some connection issue of the iPad with the monitor. I don't know why it disappeared. Just give me a second. We'll sort it out. Yeah, exit. Okay. So I was saying, in the eukaryotes we don't have the sigma subunit. Rather, we have what are known as the transcription factors, which we write as the TF. We write them as the TF. Okay, and they are numbered. The transcription factors are numbered. They are numbered from 1, 2, 3, 4, like this, or you can use the Roman numeral 1, 2, 3, 4, like that. They are numbered 1, 2, 3, 4, and so on and so forth. Now, among all the transcription factors which are there, the most important regulatory molecule is the transcription factor 2D. The transcription factor 2D is the most important regulatory molecule in the eukaryotic transcription. The transcription factor 2D is itself described to be made up of two components. First is the Tata box binding protein. In the eukaryotic promoter, we have a sequence which is just labeled as Tata box. It has a very specific sequence, and there are proteins which are able to identify this Tata box. So we simply call them the Tata box binding protein. And then we have the second set of molecules which are known as Tata box binding protein associated factor factors. Please note that this is not a single protein, but rather it is a cluster of proteins. It is not a single protein, rather it is a cluster of proteins, and that is why we use the term factors, also simply known as a TAF. So if the question asks which of the following component make up the TF2D, your answer should be TBP plus TAF. The TBP plus TAF will together make up the transcription factor 2D, which is the most important regulatory molecule in the eukaryotes. Anything else about the eukaryotic transcription you need not know. It is quite similar to the prokaryotic transcription. However, at this point you need to know certain molecules which are able to inhibit the transcription. Obviously, they will be different for the prokaryotic and the eukaryotic systems. They will be different for pro and the eukaryotic systems. Let us quickly label the inhibitors of transcription. And obviously, we'll first start with prokaryotes. Inhibitors of transcription, we'll start with the prokaryotes. In prokaryotes, the inhibition of transcription can occur in two ways. Some inhibitors are able to attach to beta subunit of the polymerase. You recall the polymerase was having the 5 subunits, 2 copies of alpha, 1 beta dash and omega. So the inhibitor can attach to the beta subunit of RNA polymerase. These include rifampicin, rifamycin, streptovaricin, streptoglycitin, and even heparin. So these are some of the molecules which attach to the beta subunit of RNA polymerase, and thereby they are able to inhibit the transcription. Then there are certain molecules which will block the DNA. So they attach to the DNA template. They attach to the DNA template. The name which you should remember here is the dactinomycin. Dactinomycin is the other name for actinomycin D. The other name for dactinomycin B, actinomycin D. So these are some of the molecules which can inhibit the transcription in the prokaryotes by different mechanisms. Majority of them attach to the beta subunit of the RNA polymerase. Some of them can block the attachment of RNA polymerase to the DNA by blocking the DNA. They can get attached to the DNA and they will prevent the interaction between the RNA polymerase and the DNA. The important name here is the dactinomycin or actinomycin D. You already know in eukaryotes, the very important inhibitor of the RNA polymerase is alpha amanitin. We had mentioned that alpha amanitin primarily blocks the RNA polymerase 2. However, it is also able to block the polymerase 1 and 3 to some extent. What you should additionally remember is from where do you get the alpha amanitin? It is obtained from the mushroom Amanita phylloides. Obtained from the mushroom, the Amanita phylloides. This is the additional point which you have to remember, from where the alpha amanitin is obtained, and it is able to inhibit the transcription in the eukaryotes primarily by blocking the RNA polymerase 2, but also it can inhibit the polymerase 1 and the polymerase 3. So that was about the transcription, and now we are moving on to translation. Any questions from the transcription? Transcription was a very simple topic. We are moving on to the translation. Any questions so far? Okay, so now we are going to be talking about the translation, and we are discussing this translation as occurring in the eukaryotes. I don't see any questions coming from you, so I am continuing with the translation as described in the eukaryotes. As I mentioned previously, it will have the similar stages as we had seen in the transcription. Actually, your question is not clear. What do you mean by source of DNA in prokaryotes? What is the question? What do you mean by source of DNA in prokaryotes? Whether it is the prokaryote or eukaryote, without DNA, we don't have the genetic material. The genetic material has to be there. So obviously, the DNA will be there inside the cell. What do you mean by source of DNA? See, the only difference between prokaryotic and eukaryotic cells when it comes to their DNA is in the eukaryotes, the DNA is present inside a compartment which is the nucleus. Whereas in prokaryotes, we don't have the membrane bound compartments, organelles are not there. So all the content is present in the cytoplasm itself. So DNA is there, but it is present in the cytoplasm. We don't have a well defined nucleus, we have a nucleoid like position where all the DNA is accumulated. This is where mostly the transcription and the replication occurs. Alright, so the only difference is that the cell bound organelles are not there, but both the prokaryotic and eukaryotic cells are going to have the DNA which was given to them during the time of cell division from their precursor cell. The DNA is one substance which always comes from the precursor DNA, and therefore in the new cell the DNA is coming from the precursor cell. I hope that answers your doubt. Okay, let's go on to translation in the eukaryotes. Again, like I told you, in the process of transcription we have three stages: the initiation. I'll be happy to take the doubt that you have if you are able to write it properly. What is the confusion in your mind? If you can mention it, then we can discuss it properly. So we have the initiation, elongation, and termination. And there is no need to say sorry. Please don't ever say sorry for asking a question. You can be putting up the question in the wrong manner, but you are always allowed to put up your question. So there is no need to say sorry. Only thing is, if you can reframe your doubt, what is the doubt in your mind, then we can discuss it out. Okay, so the translation will have three stages: the initiation, elongation, and termination. We'll take a look at them one by one, starting with the initiation. The previous two process, the replication and transcription, we had discussed in the prokaryotic cell, not in the eukaryotic cell, because in those two cells, like I said, it occurs within the nucleus inside the compartment, so it is difficult to visualize. Now you have to cross two membranes, whereas in the prokaryote you have to cross only one membrane. But the translation both in the prokaryotes and eukaryotes occurs in the cytoplasm, and therefore we will try to visualize it in the eukaryotes. Okay, moving on with the initiation. Initiation itself is discussed under multiple stages, starting with stage 1. Here what happens, the 80S ribosome, we have the 80S ribosome, it splits into two. The 80S ribosome splits into two, and obviously it will split into the components, the 60S and the 40S. It will split into the subunits, the 60S and the 40S. In the next step, in the next step, the 40s subunit will participate in certain steps, but before that, remember, the 60 and 40s we have separated, but they will have a tendency to rejoin because they have affinity for each other. So just like we had done during the replication, we bring in certain molecules attached to one of the surfaces so that the two cannot rejoin. There is something in between, so the joining will not occur. These are eukaryotic initiation factor 1A and 3. The eukaryotic initiation factor 1A and 3 prevent the rejoining of the 40S and the 60S subunits. Subsequently, the ethynyl tRNA will come in. Methynyl tRNA will come in. Alright, it will join the 40S subunit, resulting in formation of the 43S. It will result in the formation of 43S pre-initiation complex. 43S pre. Hello. 43S pre-initiation complex. Okay. This attachment between the methynyl tRNA and the 40S ribosome is facilitated by the eukaryotic initiation factor 2. Initiation factor 2 will help in the attachment of the methynyl tRNA to the 40S subunit. The next step, there is attachment of messenger RNA to the 43S initiation complex. This process is facilitated by eukaryotic initiation factor 4F. It is initiated by the eukaryotic initiation factor 4F. Please note the initiation factor 4F is made up of the subunits 4E, 4A, 4G. If you recall, I told you the 4E subunit of initiation factor 4F is able to identify the cap of messenger RNA. This we had discussed when talking about the messenger RNA and the post-transcriptional modification of the messenger RNA, where the cap, the 7-methylguanosine cap was getting attached. There we had mentioned the 4E subunit of initiation factor 4F will identify the messenger RNA via the cap during the translation. So this is where it is happening. It will identify the cap of messenger RNA and will bring in for the translation, resulting in the formation of 48S initiation complex. Resulting in formation of 48S initiation complex. So what has happened here? Everything is accumulating on the surface of the 40S subunit. See, this is the 40S subunit. So what happens? Initially, the two proteins are there, initiation factor 1A and 3. Alright. Then we bring in the methionine tRNA. We bring in the methionine tRNA. Alright. And subsequently we are trying to bring in the messenger RNA. The messenger RNA is a very big molecule. It is a significantly big molecule. So when it comes, it will quickly displace these two subunits. It will quickly displace these two subunits. So what happens? As soon as the 48S initiation complex is formed, the initiation factor 1A and 3 are thrown out. Initiation factor 1A and 3 are thrown out, and now there is nothing to stop the 60S. from rejoining with the 40s. The initiation factor 1A and 3 was preventing the joining of 40s and 60s, but now there is no one to prevent this rejoining. And in fact, this rejoining is facilitated by eukaryotic initiation factor 5, resulting in formation of ATS initiation complex. So what has happened here? We'll do a quick recap. We start with the ATS ribosomal subunit. The ATS ribosomal subunit is initially split into the 60S and the 40S subunit. To prevent the rejoining of these two subunits, the initiation factor 1A and 3 will come in and get attached to the surface of the 40S subunit. The 40S subunit will then join with the methionine tRNA with the help of initiation factor 2, resulting in formation of the 43S pre-initiation complex. To this pre-initiation complex, there is attachment of messenger RNA facilitated by the eukaryotic initiation factor 4F, resulting in formation of 48S initiation complex. Once the 48S initiation complex is formed, the initiation factor 1A and 3 are thrown out. The 60S subunit will come to rejoin, facilitated by initiation factor 5, resulting in formation of 80S initiation complex, resulting in formation of the 80S initiation complex. This is the last step of initiation. Once you get the 80S initiation complex, the initiation is complete. However, two important points to note. The formation of any amino acid tRNA, the formation of any amino acid tRNA, whether it is the methionine tRNA or the tyrosinyl tRNA or the glycinyl tRNA, any amino acid getting attached to tRNA, if you recall, we had mentioned ATP converted to AMP, it is equivalent to consumption of 2 ATP. So the methionine tRNA, which is here, represents the consumption of 2 ATP equivalent. It represents the consumption of 2 ATP equivalent. Similarly, identification of the messenger RNA and subsequent attachment to the initiation complex involves 1 ATP. Messenger RNA has been brought in and the process of identification has to be completed and then it has to be dragged in. It is a big molecule and requires 1 ATP. Lastly, when we form the ATS initiation complex, this is the messenger RNA, and on the messenger RNA, the methionine tRNA has to scan. It will scan the messenger RNA. It has to identify the correct codon and then it has to attach to that codon. Alright, all this process requires 1 GTP. So whenever, not only in this instance, whenever the tRNA attaches to the messenger RNA on the codon by the codon-anticodon interaction, it will consume 1 GTP. Remember this information will be required later. Whenever tRNA attaches to messenger RNA, it represents the consumption of 1 GTP. This means in the process of initiation, we have used up 2 ATP plus 1 ATP plus 1 GTP. GTP and ATP are equivalent. So if you want to mention the energetics in this case, it will be 3 ATP plus 1 GTP. Or we can simply write it as a 4 ATP equivalent. GTP and ATP are equivalent, so we can write it as the 4 ATP equivalent. So any of the two can be there in the option. You will have to choose accordingly what is given in the option. You will have to choose accordingly. So initiation requires 4 ATP equivalent. Initiation requires 4 ATP equivalent. Initiation is also the part of the translation which is regulated. Alright. So what is the regulation that we will get to see in initiation? The initiation factor 2 is made up of three subunits: alpha, beta, and the gamma subunits. The alpha, beta, and the gamma subunits. The alpha subunit, under suitable conditions, can undergo phosphorylation, which results in inactivation. So phosphorylation of an initiation factor 2 results in inactivation. Phosphorylation of an initiation factor 2 results in inactivation. In a similar manner, the initiation factor 4F subunit 4E also undergoes phosphorylation, but here the effect of phosphorylation is different. It causes activation. So phosphorylation of initiation factor 2, inactivation. Phosphorylation of initiation factor 4F causes the activation there under the control of various environmental stimuli as well as the hormonal stimuli. Details are not required at your level. Just remember that phosphorylation of initiation factor 2 is inactivation. Phosphorylation of initiation factor 4F results in activation of the initiation process, which is the first stage of translation. So we have completed the initiation. Let's see what is the end product that we get, meaning what is the ATS initiation complex that we have finally received. This is the messenger RNA. This is the messenger RNA that is there. On this messenger RNA, there is attachment of the ribosome. There is attachment of the ribosome. The ribosome occupies 3 codons on the messenger RNA, and you know that in each codon we have 3 nucleotides. Therefore, the ribosome occupies 9 nucleotides on the messenger RNA. The position of the codons is described in relation to the ribosome. The codons themselves do not have any name, but their position in relation to the ribosome is described. The first position is described as the E site. I'll write them one by one. The second position is described as the P site, and the third position is described as the A site. So we have the E, P, and A site. E stands for ejector site. Any tRNA which will sit at the E site will quickly be removed from the ribosome messenger RNA complex. So E is the ejector site. Once the tRNA is sitting at the E site, it will be removed. The P is the peptide site. Whatever amino acid have till now been incorporated are present as a peptide at the P site. And A is the acceptor site. This is the position where the new incoming amino acid tRNA will come and get attached. Alright, so we have the E, P, and A site. Also, don't forget that in the ATS initiation complex, we have one amino acid tRNA also. In this case, it is the methionine tRNA. It is the methionine tRNA attached to its corresponding codon, which is present at the P site. Alright, the corresponding codon which is present at the P site, if you recall, in eukaryotes, the methionine is denoted by AUG. So in the ATS complex, the codon at P site will always be AUG. Alright, in the prokaryotes is always the methionine tRNA, therefore it is AUG. In prokaryotes it is a formyl methionine, there also we have the AUG. So whether it is prokaryote or it is a eukaryote, at the P site in translation we will always have the codon AUG. So this structure that we get at the end of the process of initiation, where we had said we get the 80th gene complex, and now we are entering the phase of elongation. We are entering the phase of elongation. So what will happen? A new tRNA will come in, which has the codon corresponding to the which has anticodon corresponding to the codon at A site. So the new tRNA will come in, bringing in a new amino acid. Alright, and then it will attach to the codon. So what happened? Two things happen. Number one, entry of amino acyl tRNA. Second, attachment. Yes, the AUG is on the messenger RNA. AUG is on the messenger RNA. Attachment to messenger RNA. Rather, we should write attachment of tRNA to messenger RNA. These are the two things which have happened. These are the two things which have happened. Entry of amino acyl tRNA, and after that it attaches to the messenger. Now, once the two tRNA are in position, at this point, the pre-existing amino acid will try to form a bond with the new amino acid. The pre-existing amino acid will try to form a bond with the new amino acid, and the result is something like this. The amino acid which was there at P site, or later on it will be the peptide chain, gets attached to the amino acid which is at the A site. This reaction is facilitated by the enzyme peptidyl transferase. If you recall the peptidyl transferase, in eukaryotes it will be the 28S ribosomal RNA. In prokaryotes it will be the 23S ribosomal RNA, and they are known as ribozyme because they are having the catalytic activity. RNA with catalytic activity, they are known as ribozyme. We had mentioned this when we are doing the enzymes. So peptidyl transferase will facilitate the bond formation, and whatever pre-existing amino acid is there, whether it is single amino acid, whether it is a peptide chain, will be transferred to the new amino acid. So that's step 3. Peptide bond formation. Please note, for this reaction the peptidyl transferase does not require extra energy. ATP is not consumed in this process. All the energy is provided by the peptidyl transferase itself. Extra ATP is not required. However, as soon as the peptide bond formation is completed, there is a protein elongation factor 2 which comes into play. What it does, it pulls the ribosome forward by one codon. It pulls the ribosome forward by one codon, and this process is known as translocation. Please note, translocation requires 1 GTP. So step 4 is translocation. Step 4 is translocation. How will the translocation appear? The translocation will appear like this. the translocation will appear like this. So what I'll do, I'll try to refill that gap. Okay, right. So the ribosome moves forward by one codon. It moves forward by one codon. There's an interesting effect of this movement, and what is that? What was previously the P site, it was the second one in order, now becomes the E site. What was previously the A site now becomes the P site, and a fresh A site is exposed. Also, very interesting to note that the empty tRNA is now sitting where? It is sitting at the E site. Due to translocation, the empty tRNA is sitting at E site. And what is the function of the E site? E site is the ejector site. Any tRNA which is sitting at E site will be thrown out. So what happens in step 5? Ejection of the empty tRNA. The tRNA has already delivered its amino acid, so it is the empty tRNA. These are the 5 steps of one round of elongation, and this will keep repeating again and again and again till a stop codon is encountered. So one round of elongation includes these 5 steps: the entry of amino acid tRNA. Now we can have a fresh entry, new amino acid tRNA can come in. Once it comes in, it will attach. Once it comes in, it will attach on the A site. It is bringing in a fresh amino acid. Once the attachment occurs to the messenger RNA, there will be peptide bond formation. There will be peptide bond formation. Once the peptide bond formation occurs, there will be translocation. By one nucleotide, there will be translocation by one codon. Translocation by one codon. There will be translocation by one codon. Okay, and once the translocation has occurred, the free or the empty tRNA will be thrown out again. It will be thrown out again. And now we are ready to receive another tRNA. So this process will keep occurring again and again and again till the stop codon is encountered, and this is the phase of elongation. You can see the peptide chain will keep becoming longer and longer and longer with the arrival of every amino acid. The peptide chain will keep becoming longer and longer. Now, what happens when we have the stop codon at the A site? When we're going to have the stop codon at A site, remember there is no corresponding tRNA for the stop codon. Obviously, there is no corresponding tRNA for the stop codon. Result, no tRNA will come in. When the A site remains empty, there is entry of 2 factors. They are releasing factors. They can come in 2 combinations, either 1 and 3 or 2 and 3. They can come in 2 combinations, either 1 and 3 or 2 and 3. They will come in and they will occupy the A site. Remember, they have low affinity for A site. They don't identify any codon, they identify the A site. But as long as a high affinity tRNA is complementary they cannot get attached. Once the A site remains empty, only then the releasing factor will be able to come and get attached. So two things to note. First, we should have the stop codon. Once we have the stop codon, then the releasing factor come into play. And third thing, once the releasing factors are in place, the peptidyl transferase is able to cleave the bond between the peptide chain and the tRNA. Alright, it will cleave the bond between the peptide chain and tRNA, resulting in release of the formed peptide. The formed peptide will thus be released. It will result in release of the formed peptide. Okay, so remember, three things are required for termination. What are those three things? First, the presence of a stop codon. If stop codon is not there, the termination will not begin. Second, releasing factor should be able to come and occupy the stop codon, releasing factor 1 and 3 or releasing factor 2 and 3. Once the releasing factor is in place, peptide transferase should be available to act and will cleave the bond between peptide chain and the tRNA. The whole process requires preferably 1 GTP. If it is not there, it can make do with ETP, but preferred is the availability of GTP. So termination requires 1 GTP, or you can say 1 ATP equivalent. Alright, at this point I have a very important question which you have to tell me. Everything is in front of you, you have seen all the diagram, everything you have drawn properly. Now you have to give the answer to this question. How many ATP equivalent required for, I'm being very specific, 1 round of elongation? See, this question is very specific. I'm clearly asking you the energetics for 1 round of elongation. The options are very simple. 0, 4, 8, 9. How many ATP? 0 ATP, 4 ATP, 8 ATP or 9 ATP. What is the answer to the question? How many ATP equivalent required for 1 round of elongation? Quickly send in the answers. You're saying 1 ATP or you're saying 0 ATP? Just to recap, I'll tell you that 1 round of elongation involves the 5 steps that we had mentioned. Okay, don't forget that. Okay, second question. Suppose the options are not there and you have to give me an answer. What will be your best answer? What do you think? How many ATP are required? If the options are not there, in that case, what would be your answer? How many ATP are required for 1 round of elongation? Dr. Yashwant has given us the answer 4 ATP. Let's see. if is correct or incorrect. So these are the 5 steps of one round of elongation. If you remember the 5 steps of one round of elongation. Now, entry of amino acyl tRNA. You forgot what I told you during the initiation. Any amino acyl tRNA represents consumption of 2 ATP. This means entry of amino acyl tRNA has already consumed 2 ATP. It has already consumed 2 ATP. Attachment of tRNA to messenger RNA. We said requires 1 GTP. We specifically mentioned that the peptide bond formation doesn't require energy, but the translocation requires 1 GTP. In addition, the ejection doesn't require energy. So, what is the answer now? 2 ATP plus 2 GTP or 4 ATP equivalent. Please don't forget what we had discussed, the energetics for each step in the initiation. When you come to class 5, you cannot say that now you don't know A, B, C, D, E, F because it was taught in the nursery class. What you've learned previously has to be applied subsequently. You cannot ignore it when you encounter a problem later. You have to use that information to solve it. So the answer for one round of elongation will be 4 ATP equivalent. Now at this place the examiner can further confuse you by 2 additional questions. The question can specifically ask about addition of an amino acid, means peptide bond formation. The question says peptide bond formation requires how many ATP? At that point, if option 0 is there, that is the best answer. Why? We said the peptide bond formation does not require ATP. If option 0 is there, it is the best answer. If 0 is not there, in that case the next best answer will be 4 ATP, because in that case if 0 is not there in the option, the examiner is asking you what is the energetics for one round of elongation. So you have to be very careful about the question. Energetics for one peptide bond. The answer will depend on the options. The answer will depend on the options. If option 0 is there, best answer. If it is not there, in that case the answer will be 4 ATP. So you have to read the question carefully along with the options, and according to options you will mark your answer. If 0 ATP is there, then that is the answer. If 0 ATP is not there, then 4 ATP is answer, the energetics for one peptide bond. Because in that case the question gets converted. A recap of the translation process. Okay. We're going to do a quick recap of the translation process. We had discussed the translation in the eukaryotes. We said it occurs in the 3 stages: the initiation, the elongation, and the termination. There are 3 stages in the translation. We started off the multiple steps in the initiation. We said in step 1, the ATS will split into the 60S and the 40S. And to prevent their rejoining, we have the initiation factor 1A and 3 which joins with the 40S. The 40S will then join with the methionine tRNA under the influence of the initiation factor 2, resulting in formation of the 43S pre-initiation complex. The 43S pre-initiation complex will then join with the messenger RNA under the influence of initiation factor 4F, resulting in formation of the 48S initiation complex, to which the 60S subunit will come and join, resulting in formation of the 80S initiation complex. In the process we have discussed the 4 ATP equivalent are consumed, 2 ATP for any and every amino acid tRNA, in this case the methynyl tRNA. 1 ATP for identifying the messenger RNA and bringing it in for the translation, and 1 GTP whenever there is attachment of the tRNA with the messenger RNA. We also mentioned that the initiation factor 2 is made up of the 3 subunits alpha, beta and gamma. The alpha subunit can get phosphorylated, resulting in inactivation. Initiation factor 4F is also made up of 3 subunits 4E, 4A and 4G. 4A, 4G are always attached together. The 4E subunit can also get phosphorylated, but it results in activation. Unlike the initiation factor 2, it results in activation. So initiation ends with the formation of the ATS initiation complex. Initiation ends with the formation of the ATS initiation complex, and then we enter the stage of elongation. One round of elongation can be described as 5 steps. First, the entry of amino acid tRNA. The new amino acid is coming in. The attachment of tRNA to the messenger RNA. Subsequently, peptide bond formation under the influence of the enzyme peptidyltransferase, which is a ribozyme. The RNA are the 28 is ribosomal RNA in the eukaryotes, or the 23 is ribosomal RNA in the prokaryotes. That's step 3 of a round of elongation. And then we have the translocation, which is executed by the elongation factor 2. Elongation factor 2 will execute the translocation, and finally we have the ejection of empty tRNA. Peptide bond formation and ejection of empty tRNA do not require ATP. For all other steps, there is a specific energy component. The amino acid tRNA represents 2 ATP consumption. The attachment of tRNA to messenger RNA is 1 GTP consumption, and translocation also requires 1 GTP. So every round of elongation requires 2 ATP plus 2 GTP or 4 ATP equivalent. And in this way, the peptide chain keeps becoming longer and longer and longer. Talk about termination. Termination occurs when we have the stop codon. When we have the stop codon, the tRNA cannot come in. At that time, the releasing factor 1 and 3 will come in, or the 2 and 3 will come in and attach to the A site. Once they are in position, they are assisted by the peptidyl translocase to cleave the formed peptide. The formed peptide is then released. This is the termination of translation. Termination also requires 1 GTP preferably. GTP not there, then we can make use with ATP. So that was the translation. Any questions so far? The translation can also be inhibited by various molecules, just like the replication and transcription can be inhibited. The translation can also be inhibited. Now please note here, in eukaryotes and the prokaryotes, the structures which are participating in the translation are different. Therefore, different molecules are able to act on the eukaryotes and prokaryotes. We rarely have a molecule which is able to act on both. Very few examples are there. We'll see one of them. In prokaryotes, if you are inhibiting the translation, is it useful to us or is it harmful to us? If you inhibit the translation in prokaryotes, protein formation is blocked in the prokaryotes, it is useful to us. Such molecules will be antibiotics. Obviously, they will be covered in the pharma section. If you inhibit the translation in eukaryotes, is it useful or harmful? Obviously, it is harmful. Such molecules are known as toxins, and we are going to cover the toxins in the forensic medicine and toxicology. A little bit in the microbiology also, those toxins coming from the prokaryotes will cover in the microbiology also. So the molecules inhibiting the translation depending on the target cell can be labeled as antibiotics or toxins. We'll take a look at some examples. Let's see. Let's take a look at some examples where we will be. Yes. First, inhibitor of protein synthesis in eukaryotes. Remember, these will be toxins. Two very important molecules in this group. Number one, diphtheria toxin. Very frequently asked in exam. Some background about diphtheria toxin which you already know from microbiology. The diphtheria toxin is an exotoxin produced by Corynebacterium diphtheriae under special conditions. What is the special condition? I think we'll have to reset the screen a little bit. Let's see. We need to reset it. Yes, I need to reset it a little bit. Just give me a second. Sure. It should work. Right, resume. Okay. So inhibitors of protein synthesis in eukaryotes. We're talking about the diphtheria toxin. We said exotoxin produced by Corynebacterium diphtheriae under special conditions. It is produced only under special conditions. Only when a bacteriophage is present in the lysogenic stage. Only when a bacteriophage is present in the lysogenic stage. What are the important take-home points that you have to remember? What is the change? The change is ADP ribosylation. On which molecule? The molecule is elongation factor 2. On the molecule elongation factor 2, which is the target atom. And the target is the amino acid diphtamide. The target amino acid is the diphtamide. So 3 points to remember. The change is ADP ribosylation done on elongation factor 2, targeted on the amino acid diphtamide. Targeted on the amino acid diphtamide. Alright, so this is about the diphtheria toxin. Second, we have the ricin. Ricin is probably one of the most toxic substances known to mankind. Isolated from castor beans, surprisingly. Inactivates the 28S ribosomal RNA. Sometimes in the question, rather than 28S ribosomal RNA, the examiner can write peptidyl transferase. I'm sure you remember in eukaryotes, the 28S ribosomal RNA is actually the peptidyl transferase. What is the action of ricin? Number one, a single adenine. Write it here. A single adenine is removed. A single adenine is removed by glycolytic cleavage, whereby the action of 28S ribosomal RNA is disrupted. A single adenine is removed by cleavage. So these are the diphtheria toxin and ricin. Diphtheria toxin and ricin. On to the antibiotic. This is two examples of the toxins, how they work. The details of other toxins will be covered a little bit in forensic as well as microbiology. I will not go into those details. Let's move on to the mechanism of action of the antibiotics. There are certain other examples which I will not take. Moving on to antibiotics. So inhibitors of protein synthesis in prokaryotes are the antibiotics. One very important point to note: when I say eukaryotes, in eukaryotes we have the 80S ribosome, which splits into 60S and 40S, and the peptide transferase is the 28S ribosomal RNA. When I say prokaryotes, immediately the story changes. The story changes. In the prokaryotes, we have the 70S ribosome, which splits into 50S ribosomal RNA and the 30S ribosomal RNA, and the peptide transferase in the 23S ribosome. Okay, so remember these numbers. In eukaryotes, you will have 80S splitting into 60S and 40S, and the peptide transferase will be 28S ribosomal RNA. Okay, let's talk about prokaryotes. First, the aminoglycosides. The aminoglycosides are able to bind to either 30S or 50S depending on which aminoglycoside we are talking about. They have two important functions. They freeze initiation result, they interfere with polysome formation, and second, they cause misreading of messenger RNA code. So they are the two primary actions of aminoglycoside. They freeze initiation. Initiation, if it is not occurring, the polysome formation will not occur. What is polysome? A messenger RNA on which multiple ribosomes are attached, resulting in translation at different stages. Resulting in translation at different stages. This is the polysome. So if you block the initiation, one has got attached, the other cannot get attached. Initiation is not occurring. The polysome will not occur. The polysome will not occur. In addition, they cause misreading of messenger RNA. And then we have the tetracyclines. Tetracycle bind to the 30S and they inhibit the interaction between amino cell tRNA to the A site. They don't allow the amino cell tRNA to attach to the A site. So aminoglycoside, tetracycline. Details of the action will obviously covered in the pharma section. I'm just giving you a brief overview of the mechanism. And then we have the chloramphenicol and the macrolides. They bind to the 50S and they disrupt the peptidyl transferase activity. They disrupt the peptidyl transferase activity. Last but not the least, the erythromycin and clindamycin bind to the 50S and they hinder translocation from A site to E site. They hinder translocation from A site to the E site. Okay, so this is about the mode of action of various inhibitors which are acting on translation in the prokaryotes and in the eukaryotes. Some additional points which I want you to note. This is already done. Components of the ribosomal use subunit. I have repeatedly been saying the 80S, 70S, 60S, 50S, 40S, 30S. So what are the components of these? The 60S ribosomal subunit, you can see, obtained from 80S ribosome, 28S ribosomal RNA, the 5.8 is ribosomal RNA, the 5 is ribosomal RNA, and a large number of proteins, 50 different proteins. Going to the 40S, obviously it is smaller, it only has the 18S ribosomal RNA along with a large number of proteins. 60S and 40S from the 80S ribosome, and remember the peptidyl transferase is 28S ribosomal RNA. In the prokaryotes, from the 70S ribosome, we get the 50S and the 30S. 50S ribosomal subunit has the 23S ribosomal RNA, the 5S ribosomal RNA, and 33-35 proteins, same as for the 40S. And the 30S ribosomal subunit has the 16S ribosomal RNA and 21 different proteins. So these are the components of the ribosomal subunits, both in the prokaryotes and in the eukaryotes. Moving on, we come to the last topic, that is the lac operon model. We come to the lac operon model. First, this diagram is there. You need not draw the diagram, but we need this diagram to understand. So I'll tell you what are the components which are seen here. Lac Z, it encodes the beta galactosidase. Lac Y encodes the permease. The lac A encodes an enzyme for lactose metabolism, and lac I encodes the lac operon repressor protein. Now, why do we need to read the lac operon model? Lac operon model is basically a regulatory model. It helps to us to understand how the regulation of transcription and translation occurs in the cell. Basically, it is a regulatory model for gene expression, because gene expression involves two steps: first the transcription and then the translation. Because of the metabolism of lactose, we have a unique observation which we'll discuss subsequently. The lac operon model was chosen, and it gives us a fairly good understanding of the process. So there are a lot of points that you need to note down when you're discussing the lac operon model. So I'll keep telling you, note down this, note down this. So you must ensure that you have those points in your notes. First, the different molecules are labeled differently. This model shows us all the different types of regulation which can occur at the genetic level, which we had talked about in the enzymes. First, the lac-I repressor molecule, it represses the expression of lac-operon. If you recall, we had said induction, repression and de-repression. So the lac-I is the repressor molecule. It is a negative regulator. It represses the expression of lac-operon. Then you have lactose. Lactose is the inducer. It de-represses the lac-operon. It removes the molecule which is blocking the expression. That's why we use the term de-represses. We have some commercially similar molecules known as the gratuitous inducer. They'll remove the blockage but themselves not get metabolized. Classical example is isopropyl thioglycoside, also known as IPTG. It is the classical example of the lactose analog. And lastly, we have what is known as the positive regulator, the cap cyclic AMP regulator. It is required for optimal gene expression. So these are certain points which you need to remember. They are commonly asked which of the molecules in the lac model is the negative regulator, which of the molecules is the positive regulator, which of the molecules is the inducer. So they can ask you straightforward questions and you should be able to answer them. Okay, information about the molecules in the lac operon model. Let's move on. I'll quickly explain to you what is happening in the lac operon model and then we can write some additional points. So see what happens normally. You take a culture of bacteria and you give it two fuels simultaneously. We give it the glucose as well as lactose. So what you find, for the initial growth, the bacteria uses only glucose. Even though lactose is there, it is not used. Once glucose gets exhausted, there is a plateau phase, after which the growth starts again. But in this phase, the lactose is used. So this means the glucose, as long as it is present, will prevent the consumption of lactose. Okay, that's how and why it happens. So normally when the glucose is there, the lac-I gene will keep getting expressed. So we get the lac-I repressor. The repressor subunits will combine to form the tetramer. It goes and attaches to the promoter site. On the promoter site, we have a segment which we have labeled as the operator. So the repressor tetramer will go and attach to the operator on the promoter. So it is like a molecule attaching to the DNA template and preventing the attachment of RNA polymerase. So as long as the tetramer is there, you can see the RNA polymerase is not able to attach properly to the DNA even if the promoter site has been identified. Alright. So this is what is happening normally. This is what is happening normally when glucose is there, that is in the first phase of the cell growth. Subsequently, what happens? Glucose has been exhausted and the inducer lactose is present. See, it mentions here no glucose, no glucose, and with inducer. In this case, it is the lactose. So, when we give the inducer has more affinity for the subunits than the subunits have for themselves. So, inducer will combine with each of the members of the tetramer. As you can see, they are now separate because they are not tetramer any longer. They cannot go and block the promoter site. They cannot go and block the promoter site any longer. So the promoter site will be empty. So the RNA polymerase will try and get attached. But here there is another problem. It requires a helper to attach properly because the operator site here is narrow, and that helper is the cap cyclic AMP complex. So what happens when the glucose is exhausted? Some of the ATP and ADP is converted to cyclic AMP in the prokaryotes, and this cyclic AMP will combine with the catabolite activated proteins known as CAP and help the RNA polymerase to get attached to the DNA. Once it gets attached, then it will start doing the transcription and we get one common RNA. One continuous RNA is formed by the action of polymerase at the three different genes. Once the RNA is formed, they get transcribed into the different proteins. They get transcribed into the different proteins. So this is the lag operon model. Let's note down the other points that are commonly asked in the exam. Coordinate expression of the three enzymes concerned with lactose metabolism. Please note, the three enzymes are expressed or transcribed together. That's why we have used the term the coordinate expression of the three enzymes concerned with lactose metabolism. Coordinate expression. However, each of the protein is translated separately. If you have the coordinate expression, it doesn't mean we are going to get a big protein and then you have to cut it into multiple parts. No, it's not like that. Each protein is translated separately. This is very, very important. Lactopron will work when the special conditions are met. What are the special conditions? First, glucose should be absent. Second, the inducer lactose should be present. If these two conditions are fulfilled, only then the Lactopron model will work. And as shown in the diagram, once the glucose is exhausted, there is a lag phase before lactose metabolism starts. There is a lag phase before lactose metabolism starts. Okay, so coordinate expression, each protein translated separately, and lactopron operates when glucose is absent and the inducer, the lactose, is present. Position of the operator locus. Operator locus is placed between the promoter site and the transcription initiation site of the lag Z. Basically, it is between the promoter and the lag Z. Whereas the point for attaching the cap cyclic AP complex, it is between it is just upstream of the promoter site. Between the promoter site, between promoter site and lac I or the inducer Z. Lac I or the repressor. Lac I is the repressor gene. Lac I is the repressor gene. So it is present in between these two locations. Okay, so this is the lag operon model. At this point, we'll take a very short break. After the break, we'll do some questions and then we'll take a look at some of the techniques. So we'll take a short break. It is 9:35. Let's start again at 9:45. We'll restart. Okay, we are starting again. Um, Chalo, let's start. We're starting back up, and let us quickly take a look at some of the questions. Okay. Okay, we are starting again. Question 1. End product of purine metabolism in non-primate mammals. End product of purine metabolism in non-primate mammals is, in primate mammals, it is the uric acid. What about the non-primate mammals? What happens in the non-primate mammals? In non-primate mammals, the answer is allantoin. Conversion of uric acid to allantoin, we had said, by the enzyme uricase. And we said uricase is commercially available as the rasburicase. Very good. Very good. You're sending in the correct answers. The rasburicase. Okay. Please read the question very carefully. False regarding gout is, which of the statement is false regarding gout? Your options are due to increased metabolism of pyrimidine and due to increased metabolism of purine, uric acid levels may not be elevated, has a predilection for grade 2. Now this one all of you know is true. It has a predilection for grade 2. It is due to increased metabolism of purines, correct? Please note uric acid level at the time of the gouty attack may be within the normal range, but the uric acid and therefore gout has nothing to do with the pyrimidine metabolism, because in pyrimidine metabolism we had seen the water soluble substances A, B2C are produced. The water soluble substances A, B2C are produced in pyrimidine metabolism. It has nothing to do with uric acid and therefore gout. So the correct answer is A. A is false. A is false. Very good. The gaps between segment of DNA on the lagging strand are sealed by, means you have to join the two segments. You have to join the two segments, and the answer in this case, the answer is DNA ligase. Very straightforward question. The answer is DNA ligase. Helicase will cause unwinding. Topoisomerase removes the torsion strain. Okay. Next question. End of chromosome. End of chromosome replicated by. So end of chromosome are the telomere, and telomere are replicated by the telomerase. The telomere are replicated by the telomerase. Very good. Which enzyme has a proofreading function PCR? We have not yet done this, but I'll just like to tell you that the commonly given answer, the tag polymerase, is not correct. The answer is PFU polymerase. PFU polymerase has the proofreading function. PFU polymerase has the proofreading function. Normally, the proofreading function in PCR is not required, but where it is required, we have to make use of the PFU polymerase. Telomerase are not active in. Now, telomerase means cells which have to divide unlimited number of times. So germ cell have to divide, hematopoietic cell have to divide, cancer cells have to divide, but not the somatic cell. The answer is the somatic cell. The answer is the somatic cell. And the somatic cell will follow what is known as the Hayflick's limit. This we have discussed. For humans, the Hayflick's limit is about 70 divisions. Okazaki fragments are produced during synthesis of single-stranded DNA, double-stranded DNA, messenger RNA, or tRNA. What is the correct answer? Now, in this case, the correct answer is double-stranded DNA. When the DNA polymerase has to transcribe two anti-parallel segments, only then we get the Okazaki fragments. I can see all of you are giving mostly the correct answer, so it's very good to see that you have understood the underlying concepts for the molecular biology. Now we discuss this. For one peptide bond formation, how many high energy phosphates are required? If option 0 is not there, in that case our answer becomes 4 ATP. If 0 were there, then it will be 0. If 0 is not there, in that case the answer will be 4 ATP. Now this is a slightly tricky question. All of the following are involved in microRNA synthesis except we know that Drosha is involved, we know Dicer is involved, we know that the DGCR8 is known as Pasha, so Sasha is not involved. Sasha is not involved. So the correct answer is Sasha. Sasha sounds like a villain of some Hindi serial. Those Hindi serials which are not coming nowadays, they have some villains like Sasha and Pasha. Okay, so at this point there's some other things that I want to talk to you about, and first and foremost in that list is the polymerase chain reaction. Before the polymerase chain reaction, let me see. Yeah. Yeah. Okay. Now, very frequently we get questions on techniques related to the separation of proteins. So we'll take a quick look at the fundamentals, very basics of the techniques for separation of protein. Obviously, in this class we cannot have the detailed discussion of the techniques for separation of proteins. So we'll quickly take a look at the various methods which are used for the separation of proteins. Previously, right in the beginning, when the study of the proteins was initiated, for separation of proteins, we used the property of solubility. Even in the UG lab, we do this separation by precipitation by heavy metals and salting out. Salting out is commonly done to separate the globulins and albumin from plasma. The globulins are precipitated at half saturation. Albumin is precipitated at full saturation with ammonium chloride with the salt. Then we shifted to separation on the basis of molecular size. For example, dialysis in patients, we are doing it on the basis of molecular size. The gel filtration chromatography. The gel filtration chromatography, also known as the size exclusion chromatography. Gel filtration chromatography, also known as the size exclusion chromatography. And lastly, we can have the ultracentrifugation. The size exclusion chromatography is special, or the gel filtration chromatography is special, because in this case the largest particle comes out first. So this is a unique method of the chromatography. Normally what happens in chromatography, the smaller particles will come first, then the bigger than that, and the more big particles, and lastly the biggest particle will come. But in size filtration chromatography or the gel filtration chromatography, the largest particle comes out first. So this is the point you have to remember. The largest particle comes out first. Most commonly nowadays we separate the proteins on the basis of their molecular charge. There are a large number of mechanisms by which we can do this. The ion exchange chromatography. The ion exchange chromatography can be based on positive as well as the negative beads depending on what is your target. Depending on what is your target, we can have the positive and negative beads for the ion exchange chromatography. Same for HPLC, high performance liquid chromatography, the electrophorus, the page, the isoelectric focusing, and SDS page. All of these will carry out the separation of protein on the basis of molecular charge. A very precise separation. See, all of them have very good separation but not exquisitely precise. But there are certain techniques by which we can get an absolute pure form of the protein. Two of them include the affinity chromatography, very specific, and precipitation by antibodies. Precipitation by antibodies, the principle is somewhat similar to ELISA, but there are some differences. In these two mechanisms. We get the extremely pure form of protein. The extremely pure form of protein is obtained. Please note, very frequently you'll get the question on these techniques, one or two line that you should know, the basic fundamental you should know. The separation is done on the basis of molecular charge. The charge is the driving force. However, the separation can occur along different parameters. The separation can occur along different parameters. For example, in SDS page, the separation occurs as per mass. In isoelectric focusing, separation occurs as per PI. You know what is PI? Isoelectric pH. Alright. So these are but just few instances where the criteria of separation may be different, although the driving force is the charge. The driving force is the charge, but the criteria of separation can be different. Okay, so these were some important points related to the protein separation. Now let's quickly talk about the polymerase chain reaction. I am sure all of you know what is polymerase chain reaction. The polymerase chain reaction is used for amplification. It is used for amplifying DNA. It is used for amplifying DNA. Right? So for the polymerase chain reaction, you should know two things. Number one, what are the substrates that will require for the polymerase chain reaction? And number two, what are the steps of the polymerase chain reaction? So coming to substrates, you must have the template DNA for which you want to do the polymerase chain reaction. If template DNA is not there, then you cannot work. It is the basis on which the new DNA will be formed. To identify the segment in this DNA template which you want to carry out for polymerase for PCR, you must be able to identify a certain segment. So you must have the primers. Once you have these two, then you will require the enzyme which will participate in the polymerase chain reaction. Commonly it is the Taq polymerase, but it is not necessary to always use the Taq polymerase. For example, I've given you the example where we may use the PFU polymerase. To make the new DNA, we require the building blocks.
which are the deoxynucleotides. The deoxynucleotides are required as the building block. Magnesium is required as the cofactor for the polymerase, and the pH must be maintained at the same value, and therefore we require a very good buffer to maintain the pH at the same value. These are the substrates required for a polymerase chain reaction. What are the steps required for the polymerase chain reaction? You'll note I write step 0. Means if you don't carry out this step, the rest of the steps in polymerase chain reaction do not make any sense. What is just step 0? It is based on the primers which are required to demarcate the segment of DNA. So you have to design the primers and then you have to synthesize them. If you are not able to complete this successfully, the rest of the PCR doesn't make any sense. You have to complete this successfully. Once we have the primers, once we've designed and synthesized the primers and we have all the substrates, we carry out the separation of the two strands, which is known as denaturation. After separating the two strands, we allow the primer to attach. This is known as annealing. Once the primer is attached, then we allow the primer to get extended to give the final DNA, and we call it the elongation or the extension. Step 1, 2 and 3 are repeated n number of times. 1, 2 and 3 make up one cycle, and typically we have 35 to 40 cycles. Typically we have 35 to 40 cycles before we wait to analyze the final result. Please note, after completing the required number of cycles, we have what is known as a long elongation phase. The time frame for each is slightly different. The denaturation occurring at 94 degrees Celsius for about 15 seconds. Time frame may be same. Annealing occurs in the range of 45 to 65 degrees Celsius. Time required 15 to 30 seconds. The elongation or extension for the Taq polymerase is carried out 72 degrees Celsius. It requires 30 to 45 seconds. However, the elongation phase also occurring at 72 degrees Celsius, but typically it runs in minutes, 5 to 10 minutes. 5 to 10 minutes required for the long elongation phase. This ensures that incompletely formed DNA fragments are not available because they will make our interpretation very difficult. So at a very basic level, for the polymerase chain reaction we require the 6 substrates which you have listed, and a series of steps need to be performed. At the end of these steps we still don't know the result. Electrophoresis. Only when we complete the electrophoresis we'll get to know whether the successful polymerase chain reaction has occurred or not. Typically the dye used for the DNA gel electrophoresis happens to be the ethidium bromide. Because of the carcinogenic potential of ethidium bromide, nowadays it is being replaced by SYBR green. Because the carcinogenic potential of ethidium bromide, nowadays it is being replaced by the SYBR green. Now there's a problem here. once you have to, first you have to complete the PCR, and after you do complete the PCR, then you have to perform the DNA gel electrophoresis. There is no way that even in between you get to know whether the PCR has occurred successfully or not. So here we can design our process in a slightly different manner. What we do, to the mixture we add certain certain dyes. We are using special dyes. These dyes have a very unique property. They fluoresce only when attached to DNA. We are using some special dyes which fluoresce only when attached to DNA. Examples include SYBR green, Cyto 9, LC green, and Eva green. These are some of the dyes which will fluoresce only when attached to DNA. There are some other dyes which are sequence specific, so we are not going into those dyes. We focus on the dyes which are generic and they fluoresce when attached to DNA. So what we do, we put some of these dyes in the mixture of the substrate mixture. We put some of these dyes. So normally they are not going to give any significant fluorescence. We have a sensor here. And then we add the reagents and allow the reaction to complete. We allow the reaction to complete. Please note I am drawing the DNA with green color, new DNA. So initially what happens, some of the dyes are attached to these DNA particles and a background fluorescence is obtained. Background fluorescence obtained even when before we have started the PCR. And now after every cycle, certain amount of new PCR will be formed, and therefore the fluorescence in the mixture will start increasing. After second cycle, each of them will synthesize a copy, and now the fluorescence will also commensurately increase. So if you find the increase in fluorescence which is identified by the sensor, if you find the increase in fluorescence which is identified by the sensor, even in between we can say that the polymerase chain reaction is successful. So what we are doing here, we are monitoring PCR in real time. We call it the real time PCR. Very important aspect to note here is, if the PCR is successful, we get to know whether it is successful or not within 20 to 25 cycles. Typically we draw a graph like this. Here we have time and here we have the measurement of fluorescence. So initially we get the basal fluorescence, baseline fluorescence is there. As the reaction becomes successful, there is increase in the fluorescence. However, after some time all the dye will be occupied, so we reach the plateau phase. So what we are doing, we are monitoring the increase in fluorescence over time, and this helps to tell us whether the PCR has been successful or not. Because we are able to monitor the progress in real time, this condition is known as real time PCR. If you run the real time PCR with standards, it works as quantitative PCR. We can quantify the starting amount of DNA in a given sample when we run the real-time PCR with standards. That is why it is known as the quantitative PCR. We have another very important point which is known as isothermal PCR. About isothermal PCR, what you need to remember is the role of BST polymerase. The BST stands for Bacillus stearothermophilus. It stands for the Bacillus stearothermophilus, and the polymerase from this has a very unique property. First and foremost, it will carry out polymerization just like any other polymerase. It will carry out polymerization just like any other polymerase. But at the same time, it also acts as helicase. So it is a dual function polymerase. It is a dual function polymerase, and it can be used for what is known as isothermal PCR. Please note, isothermal PCR is now available in the market. Commonly, the enzyme which is used here is the Bacillus stearothermus polymerase. The enzyme obtained from Bacillus stearothermophilus. This enzyme has a unique property of carrying out the additional unwinding of the DNA. It is able to carry out the unwinding of the DNA. All of these are mechanisms for amplification of DNA. What is the mechanism for amplification of RNA? A technique for amplification of RNA. The RNA amplification can be done by the technique called NASBA. By the technique called NASBA. NASBA stands for nucleic acid sequence based amplification. NASBA stands for nucleic acid sequence based amplification. Please note, this technique is also isothermal in nature and the yield is more than PCR. So NASBA is an isothermal technique for RNA amplification. This is also isothermal in nature. Okay, amplification is good, but analysis of the RNA is difficult. Why? Because RNA has a very short half-life. It is very fragile. So identifying or detecting the RNA after amplification is somewhat risky because the RNA will quickly degrade. So how do you identify or quantify the RNA? For this, what we do is first convert the RNA into what is known as the complementary DNA. It is converted into complementary DNA, and the complementary DNA is then subjected to PCR. The conversion of RNA to cDNA is known as a reverse transcription, commonly written as RT, reverse transcription or RT. And this, as you can see, is followed by PCR. This PCR may also be the real-time PCR, it may be the normal PCR. So when you convert the RNA into cDNA and then subject the cDNA to the polymerase chain reaction, the whole process is known as RT-PCR. The whole process is known as RT-PCR, the reverse transcriptase PCR. The reverse transcriptase PCR. Reverse transcription PCR. The PCR which is being used can be the traditional PCR or it can be the quantitative or the real-time PCR. Any of them can be used. The reverse transcription PCR will be used for viruses with RNA genome. It is used for making the diagnosis of viruses with RNA genome. and it is very likely to land up in your exam. It is being used for identification of COVID-19 infection. You remember that COVID-19 is being caused by the novel coronavirus type 2 virus, which is the RNA virus. So the reverse transcription PCR is the technique which is being used for identification of COVID-19 infection. Very likely to land up in your exam. Very likely to land up in your exam. So this is about the different variants of nucleic acid amplification. Traditional is the PCR, then we can have the real-time PCR, then we have the isothermal RNA amplification, that is NASBA, and lastly we have the reverse transcription PCR, which is very important. If you use the quantitative PCR, then you can also label the reverse transcription PCR as QRT PCR, or you can write the full name RT real time PCR. RT is reverse transcription, and then you can write the real time PCR. Okay, this is about the polymerase chain reaction that you must know. There are few other terms that I want you to introduce to. First is FISH. FISH stands for fluorescent in situ hybridization. What it does? Identifies nucleic acid by hybridization, and here we use a probe with fluorescent tag. We use a probe with fluorescent tag. Okay, so fish is compared and contrasted with ELISA. In ELISA we have the antigen antibody reaction, all right, and it is tagged with enzyme. Here what do you see? Here we see the hybridization. Hybridization means the pairing up between nucleic acid, and the tagging is done with a fluorescent dye. Tagging is done with a fluorescent dye. The probe is tagged with a fluorescent dye. What do you use the fish for? Fish can be used to identify specific allele of chromosome. If you synthesize the probe in such a way that is specific for a particular allele, so you can use it to identify specific allele of chromosome. Then commonly it is used for karyotyping, whether one or more chromosome or a segment of chromosome is missing. Lastly, the probe may be targeted towards RNA, where it can be used to study gene expression. Although there are cheaper ways to do that, theoretically we can use it to study the gene expression. So this is the fluorescent in situ hybridization. Then we have what is known as FRAP. FRAP stands for fluorescent recovery, rather it is fluorescence recovery after photo bleaching. Without going into the details, what you have to remember, what is the purpose of FRAP? Why do we use the FRAP? To identify protein movement in membrane. In this way, we are able to demarcate which are the integral proteins and which are the soluble proteins, because the integral protein will generally not move, it will remain fixed at a particular location, but the soluble proteins are generally liable to move along the cell membrane. So that is the frap. So these are some of the important additional techniques which are commonly asked. Every time there can be some new techniques, so you need not worry about the new techniques because everyone else will be in the same boat. Okay, one more thing that I want to talk about is, this is likely to be covered in your pathology section also, the flow cytometry, but this is one thing which I wanted to talk about, the flow cytometry. The flow cytometry has a very simple principle. It is based on flow of cells in a single channel. So what we do, we label the cells and then we pass it through a channel where one cell can go at a particular time. One cell can go at a particular time. The types of probes that you can use are two: antibodies tagged with fluorescence. Very commonly we use this, the antibody tagged with fluorescence. The alternative is we use antibody tagged with magnetic beads. Now what is the difference between the two? When the antibody is tagged with fluorescence, we use it for determining the absolute number as well as relative number of a cell type. For example, you can use it for the CD4 CD8 ratio, or the absolute number of CD4 CD8 cell count, absolute number or the ratio of CD4 to CD8. But when we are using the antibody tagged with magnetic beads, this is used for separating the different cells. Different types of cells can be separated. We call it the cell sorting. Cell sorting is one of the preliminary steps to be done before what is known as gene therapy. All right, even before the, it can be used for the cell cultures. You want to grow a certain type of cell, so you have to separate it. And how do you separate it? We separate it by the cell sorting. So remember, the flow cytometry can involve antibodies of two types. Either you can tag them with fluorescence or you can tag the antibodies with magnetic bead. The purpose of the two tagging is different. If you tag it with fluorescence, then what we are doing is determining the number and the type of cells. But if you tag it with magnetic beads, we use it for separation of the cells, and this is known as the cell sorting. So we can have the cell counting and cell sorting. Cell sorting can be a precursor for gene therapy or the cell culture. It can be the precursor for gene therapy or the cell culture. Okay, so these were some of the very important techniques which sometimes land up in your NEET exam. Uh, Although every time they give some new technique about which nobody will be knowing anything, so no need to worry about it. However, if it is one of the older questions, you must be able to answer it. That's why I've told you some of the very important techniques which have been asked previously. At this point, we are closing our session on biochemistry. I hope you have had a very good overview of biochemistry and you will feel confident in solving the questions of biochemistry from now on. One very important word of caution: please don't solve very old question banks, because in the old question banks the questions which are there are no longer being asked in your exam. Since the NEET and the next NEET exam has come in, there has been a change in the focus of the examiners. So it is a very good idea to focus on the questions from the last 6-7 years only. At most you should do the last 10 years. Don't go beyond the last 10 years. Your focus should be doing the questions last 10 years. Preferably, because of a limited time, you can limit yourself to the last 7 years. If you are able to do questions from 2013 onwards, that should be more than enough. It will hold you in good stead, and you need not go beyond 2013. If you have time, obviously you can go further, but even then, don't go beyond 2010 because of change in focus of the examiners. Nowadays, mostly we get questions on the clinical part, or the basics which are related to clinical part, or basics which are involved with topic of research. So that is the criteria for selection of questions from biochemistry. Plus, always keep in mind that if you have a good command on biochemistry, then it has certain overlap with the pathology, and certain overlap with the inborn error of metabolism that we see in the neonatology section, or the pediatric section. So okay, iron. Okay, let me see. We have the slide for iron. Okay, we will do the iron. Just give me 1 second. Yeah. Just give me 1 second. The screen has gone off. Just give me 1 second. What happened to the screen? Okay. Oh, what happened? Okay, sorry for the loss of the screen. Okay, so let us quickly talk about iron and the iron transport. So iron, is a very important metal. It is required for a large number of proteins inside our body, most important of which is the heme. And besides the heme, it is also required as a cofactor for a large number of proteins. For example, when we're doing the TCA cycle, there we had seen that iron is required by the aconitase. So it is very frequently required by a large number of enzymes. So let us quickly take a look at the iron. Mostly what you need to know is the transport which is occurring for the iron. Okay, so in the iron transport there are two things to keep in mind. First foremost is the absorption of iron occurs differently depending on the source. We can we can have the heme iron, and we can have the non-heme iron, meaning the iron which is coming from the plant source. Okay, the uptake of heme iron is independent of the iron coming from the plant source. And you can see we have a separate transporters for the heme iron. At the lower part you can see we have the separate transporter for the heme iron, the SLC46A1, also known as SCP1. So it is the heme iron transported separately. It goes into the enterocytes, and in the enterocytes it is then broken up. The iron part is separated, and the heme part will then be broken down independently, as we had discussed previously how the heme part is going to break down. Okay, and this iron will be converted to the ferritin, and the ferritin is the storage form. Subsequently, it can be transported to the cytoplasm, from where it can be taken to the tissues. Okay, what about the non-heme iron? The non-heme iron has to be absorbed in the ferrous form. As you can see, we have mentioned the ferrous form. In the top we have the ferrous form. The pen is not working. Okay, so this is the role of vitamin C in the absorption of iron. The iron in the diet is typically in the ferric state, Fe3 plus state, and it is converted to the ferrous state by the reducing substances. It can be the acid, and most commonly vitamin C helps in the absorption of iron by converting it to the ferrous form. The ferrous iron is quickly absorbed by the divalent metal transporter. Please note that DMT1 is non-specific. It will act for the calcium, it will act for magnesium, it will act for iron. All the divalent metals are transported by the same transporter, the DMT1. Okay, so two things happen. First, the ferric iron is converted to ferrous iron by the DCYTB, and then the ferrous iron is taken up by the DMT1. Alright, so the ferrous iron is now inside the cell. It is transported to the blood. Now look on the other side, it is transported to the blood by the ferroportin. The transporter is the ferroportin, and when it is being transported outside, it is converted into the ferric form. The hephaestin will convert it into the ferric form, and this ferric iron is transported by a transferrin. So I'll summarize the points that you need to know. Number one, separate transport for heme iron and the non-heme iron. Heme iron is readily taken up, so the availability of heme iron is better. Even if you consume less amount, more of it will be absorbed. The non-heme iron is transported separately. Its absorption is poor, so you need more of non-heme iron to compensate for the defect in the limitation in the transport. Number two, the non-heme iron must be present in the ferrous form. However, when we consume it, it is there in the ferric form, so it has to be converted to ferrous form, which is facilitated by the presence of vitamin C. Once the ferrous form is formed, then it is transported by the DMT1. The limitation here is DMT1 will transport all the divalent metals. Therefore, there is competition between calcium, magnesium, iron. All of them will compete, and limited absorption occurs. There is another reason why when you consume the iron tablet, when you give the iron supplement, you have to ensure that the individuals do not consume maybe calcium rich food, that is milk or milk products, or certain chelators which can block the absorption of iron, for example phytates. So you cannot combine the consumption of iron with those substances. In addition, you will try to club it with vitamin C, because it will maintain the iron in the Fe2 plus, and absorption will be better. From enterocyte, the iron will be transported into the blood, and where it is oxidized, The oxidation can also occur by other molecules, and the Fe3 form is transported by transferrin. The transferrin is the transporter in the blood. So these are some points about the iron metabolism that you should know. In your books, there may be a discussion on various other minerals, but they are generally not required. What do you need to know about homocystinuria? We have a question called homocystinuria. I have not covered it because it is covered separately in medicine when we are talking about the cardiovascular disorders. There we talk a little bit about the homocystinuria. That's why I have not taken up. Porphyrias, I can show the porphyrias again. Let me see the table for porphyrias. I'll show you, so it will give you a good picture of what we had discussed. Just give me a second. Porphyrias. Porphyrias, where are the porphyrias? Just give me a second. Monosaccharide, yeah, we've reached the porphyrias. Yeah, okay. So this is the list of porphyrias. In the meantime, Murtaza, can you specify anything you want to know specifically about the homocystinuria? Then we can take it up. This table shows the important porphyrias. You will note the enzymes are labeled in sequence: enzyme number 3, 4, 5, 6, 7, and 8. If you recall, in the heme synthesis we had said there are 8 reactions. Enzyme 1 was the ALA synthase, enzyme 2 was the ALA dehydratase, which are not listed here. Reason being, the old identification of porphyria I told you was based on Ehrlich test. The old classification of identification of porphyria was based on Ehrlich test. The Ehrlich test identifies, what does it identify? It identifies porphobilinogen. The Ehrlich test identifies PBG. The PBG, if you recall, is formed by the ALA dehydratase, right? So until unless the PBG is formed, the early childhood test will not become positive, and therefore the old identification of porphyria starts with enzyme number 3. Newer classification will also list enzyme 1 and 2 in the list of porphyria. It will also list the enzyme 1 and 2 in the porphyria. Now here, what are the things that you need to know? Number 1, the photosensitivity is not seen in acute intermittent porphyria. You can see in all the others you find the photosensitivity, but it is not seen in acute intermittent porphyria because ring structure is not formed here. If the uroporphyrinogen synthase 1 is not there, the porphobilinogen will not be formed. If porphobilinogen is not formed, the ring structure will not be formed, and the cause for photosensitivity is the ring structure. So as long as the ring structure is not there, we don't find photosensitivity in acute intermittent porphyria. In addition, the porphyrias which are erythropoietic in nature, protoporphyria, congenital erythropoietic porphyria, they do not have abdominal pain. The abdominal pain is there in all others. It is there in all others, but it will not be there in the erythropoietic porphyria. The abdominal pain may or may not be there in Porphyria cutanea tarda. I had mentioned that nowadays the identification of the porphyrias is with the help of HPLC. It is done with the help of HPLC. HPLC can directly identify the type of porphyria. Now there are two porphyrias for which you must remember the names of the enzymes. Two porphyrias for which you must remember the names of the enzymes. The two most common porphyrias, that is the acute intermittent porphyria, enzyme defective deficient is the uroporphyrinogen 1 synthase, and next the porphyria cutanea tarda, where the deficient enzyme is the uroporphyrinogen D carboxylase. Uroporphyrinogen D carboxylase. So these are the two porphyrias for which you must remember the name of the enzyme. In addition, I mentioned that some common features. I told you about the test that we performed, the early childhood test and the HPLC, and we had also talked about something known as the Soret band. So these are some of the points that you should know about the porphyrias. In addition, remember the iron will interfere with the formation of heme. Iron will interfere with the formation of heme, and intermediates of heme synthesis may be precipitated in that condition. Murtaza, I don't have a ready list for homocystinuria at this time, unfortunately. But what I'll do is I'll send across the list for homocystinuria to your course coordinator, Dr. Manoj and Dr. Shravan. I'll hand them over to them, which they will share with you. I unfortunately today I don't have the slide for homocystinuria. So I'll send it across to Dr. Manoj and Dr. Shravan, who will share it with you. If there is any other thing that you want me to take up, please let me know. Any other questions you'll want me to take up, then please let me know. The homocystinuria part, I will ensure that you get the appropriate notes for that at your convenience. Any other question? Any other question? Any other question? All right, so okay, that's a tall order. Let's see what we can do about it. We'll try to link the uh, metabolic cycles. Let's see. Okay, so typically we start with glucose. When we are talking about linking the metabolic cycles, we start with glucose. The glucose will get converted to glucose 6 phosphate, right? The glucose 6 phosphate in turn can go towards glycogen metabolism. The glucose 6 phosphate can go towards HMP shunt. Glucose 6 phosphate can go towards the HMP shunt. All right. Now, subsequently, the glucose 6 phosphate via multiple reactions will go to the pyruvate. Via multiple reactions will go to the pyruvate. Now, this pyruvate interchangeably links with 5 different amino acids. Interchangeably, it links with 5 different amino acids. In addition, via the mitochondria. Via the mitochondria, it goes back towards the gluconeogenesis. The pyruvate gets converted to oxaloacetate, and it also gets converted into acetyl coenzyme A. The oxaloacetate was the part which was going to the gluconeogenesis. The oxaloacetate is also linked to aspartate, which in turn is linked to several amino acids. The fatty acids will enter the mitochondria by carnitine transport. They will enter the mitochondria by carnitine transport, and here the fatty acids will get converted to acetyl coenzyme A by beta oxidation. By beta oxidation, the fatty acids get converted to acetyl coenzyme A, and this acetyl coenzyme A subsequently gets metabolized via the Krebs cycle. The Krebs cycle is again linked at multiple places with the amino acids. It is linked at multiple places with the amino acid. In addition, the citrate which is formed here, the citrate can come out. And get converted into oxaloacetate plus acetyl coenzyme A. Can get converted to oxaloacetate plus acetyl coenzyme A. The oxaloacetate goes back into the mitochondria, but this acetyl coenzyme A can participate in fatty acid synthesis and cholesterol synthesis. The citrate in turn is able to regulate the glycolysis. It is able to regulate the glycolysis. It also promotes the fatty acid synthesis. Alright, so the proteins via the amino acid enter the metabolism at the level of Krebs cycle or at the level of pyruvate. Either the level of pyruvate or at the level of Krebs cycle. Where they enter the Krebs cycle, that particular reaction is known as the anaplerotic reaction. Please note, at the same time, these reactions are also amphibolic. anabiotic reaction, whereby we are interconverting the amino acids into the intermediates of the Krebs cycle. That is the anabiotic reaction. Because these reactions are bidirectional, they are also known as the amphibolic reactions. So this is how we are briefly linking the various processes. You can see all of them are interlinked. The glucose via the metabolism goes up to the pyruvate and then enters the Krebs cycle. The Krebs cycle can be directed towards synthesis of amino acid when we have excess of glucose. When the glucose is less, the amino acid can enter the Krebs cycle and they can be directed towards the gluconeogenesis. So this is the interlink between the amino acids and the glucose. Similarly, when there is excess of glucose, a part of it can be directed towards fatty acid synthesis as well as cholesterol synthesis. But when glucose is less, the fatty acid will enter inside and it will enter the Krebs cycle and it will give us the energy. In addition, the acetyl coenzyme which was formed in the mitochondria, when it is coming from fatty acid, it can also be directed towards the ketogenesis. It can also be directed towards ketogenesis. So in turn, the various fuels, you can see the ketone bodies, the fatty acid and the glucose, they are all interlinked. Lastly, the fatty acid and the ketone body metabolism can only occur when there is at least some amount of glucose, because if the glucose is not there, from where are you going to get the oxaloacetate? Oxaloacetate comes from pyruvate. So some amount of glucose should always be there to metabolize the fatty acid as well as the ketone bodies. Okay, any other question? So if there are no more questions, I think we'll call it a day. Please feel free to contact me if you have any doubts or if you have any questions. You can drop me a message at I'm available on Facebook, or you can drop me a message on Telegram or WhatsApp. So you can contact me through any of these. I'm always available to you. Please don't hesitate if you have any questions whatsoever. You can use any of these platforms to connect with me. Thank you very much. Please take care, and I hope these sessions were useful to you. And I'm pretty sure that you will be able to solve almost all the questions which are coming in the exams from what we have discussed over the last few days. We have had quite an exhaustive discussion, and we